Rui Benfeitas, and I'm a bioinformatician at the National Bioinformatics Infrastructure in Sweden. And I would like to welcome you to this tutorial together with my colleagues, Nikolay and Ash. And so that we can get started, I will share my screen and hopefully you will be able to see my presentation. All right, so I hope everyone can see my screen now. So before we get started, just to lay down a few rules of what we will follow today and tomorrow. So I would ask you to please remain muted. And if you would like to ask a question during the lectures, you can do so, but please raise your hand before you want to ask a question, as we will be probably over 40 in this session. Perhaps we will not have enough time to take all the questions during the lectures. And if this is the case, please, you will use the HackMD link that one of us will provide you over chat very soon. And it's also in the course website that you've received. We would like to ask you, if you can, to please keep your cameras on so that we don't feel like we are speaking alone. And just be noted that the entire tutorial will be recorded. So as I've mentioned, my name is Rui Benfeitas. I'm one of the organizers of this tutorial together with Nikolay and Ash. You will be hearing from Nikolay today and from myself and Ash tomorrow, but the three of us will be with you throughout the entire time. And we are all senior bioinformaticians at the National Bioinformatics Infrastructure Sweden in Science for Life laboratory, located both in Stockholm and Lund. Because we are a bit too many for us to introduce ourselves, I would like to ask you to please go to Menti, to this link that one of us will share with you on the chat, so that you can at least tell us a few things about yourselves. So we can do it in this way. My apologies for stopping to share. We will take it again. So we see that 9 people have voted. We would expect that most of us would come from Sweden, since this tutorial is linked with the European Conference in Computational Biology, but we see that we have some participants from the US, it seems. Let's see if we get any more votes. Some from South America. Oh, wow, we have someone quite far away. All right, so perhaps we can take the next one. And I am having some difficulties here. Okay, so it would be interesting for us to know also what is your experience in terms of omics. So what type of data have you used in your research or are you currently using? So you can now select multiple choices if you use more than one data set. Very interesting to see. So most people coming from a transcriptomic perspective, both for bulk and single cell, that aligns well with what we will be talking about. A big part of the data integration that we will be discussing is about RNA-seq or proteomics as well. We have seen some recent interest in microbiome data analysis and its integration with other sorts of omics, but this is something that we do not touch in here. So we have 25 answers from 32 participants. All right. But I think this gives us a good idea of what most of us are working with. And we can go back to this one. All right, so what we will do during the following days is we will be talking about different integrative methods and network analysis. I hope that all of you have been able to go through the data preparation, the course preparation materials, which included the data preprocessing, the feature selection, and the dimensionality reduction notebooks. So today we will be hearing about machine learning view of integrative omics with a more detailed focus in supervised and unsupervised integration, after which we will follow this with a 1 hour, a bit over 1 hour hands-on session, and end the day with a short summary and recap of that type of the hands-on session. Tomorrow we will hear about biological network analysis and the application of network analysis in meta-analysis. And then we will also follow these lectures with hands-on sessions and a short summary. So I understand that some of you may have some questions in terms of installation, but what we will focus these hands-on sessions is not on solving any particular installation issues or programming questions. We will focus these two hands-on sessions on a more scientific discussion. So we will give you details on how we will organize these hands-on sessions later on today. As you already have the course homepage, all the information should be there. So all the slides and scripts, you can access them from the schedule page. The hands-on instructions you can find from the labs page, but if you have not been able to install everything in your system, that's fine. You can still take the HTML files and follow get the overall feeling of what you should be paying attention. throughout. As I mentioned, time permitting, we will take questions during the lecture, so feel free to raise your hands here on Zoom. If you have a question that you would like to ask, that's perfectly fine. If you prefer, you can instead ask them on HackMD. And at the end, we will ask for your feedback. So on HackMD, you will see, for those of you that have never used it, perhaps when you click the link, you will see the website like this. But for you to ask questions, you need to go here on the top left corner. You click, and then you can edit your question and insert your question here. So feel free to edit as you wish. Just a final note. So this tutorial is a shortened version of a longer workshop that we run. And we actually have applications open for this workshop. It will take place from 6th to 10th of September. And the applications are open to researchers from European institutions. And this is a free of charge workshop. So if you are interested, please go to the course homepage. I will share it shortly over chat, but please consider applying or share with your contacts if you feel that this can be useful to them. All right. Any questions so far? If not, then I will give the word to Nikolay.
Thank you very much, Rui. Hello, everyone, and welcome to this tutorial number 4 within ISMB ECCB Conference 2021. This is a tutorial number 4, a practical introduction to multi-omics integration and network analysis. My name is Nikolay Oskolkov, and I'm from the National Bioinformatics Infrastructure, Sweden. And in this lecture, I'll be covering some aspects of omics integration, particularly machine learning aspects of omics integration, which is one direction in data integration. It's not the only direction. There are different approaches. What I'm going to talk about is not sort of written in stone. People do it in different ways. I'll be concentrating on how we can use concepts from machine learning to better understand and evaluate and define data integration. And I'll be assuming that you guys have a more or less biological background. So you're doing some data analysis related to biological data. Yes, so basically, if you go beyond biological data and look around us, so we have the Internet of Things, which is full of data. It can be tabular data, it can be images, it can be time series data or text. If you look inside the data, they always have something to do. You can always boil it down to numbers, to matrices or high dimensional arrays. And you can conceptualize these numbers as data distributions. It's useful to talk about data and to understand data, looking at data distributions. And all these data types, such as tabular, image, or text, happen to come to very different data distributions. So it would be very naive to just merge them together without taking into account this difference in their distributions. So basically, that's pretty much the challenge and one of the goals of data integration to understand how we can combine multiple data types in order to have some synergistic effects at the ends. So typically, when you work with data, you very often can visualize your data as a matrix, as a data matrix. And this data matrix can have two dimensions, or has two dimensions. It's a n dimension and p dimension. I will be calling them n and p dimensions. The n dimension is a dimension of statistical observations. So it's a samples or cells. So you happen to observe expression of a gene for one sample, two samples, three samples, etc. And the p dimension is a so-called feature dimension. So P can be genes, can be proteins, can be nucleotides, or maybe pixels if you're working with images. And for each P, for each feature P, you can have n observations. So when you combine different data types together, when you put different omics, biological data together, you usually has this kind of situation. You increase the dimension P, but you keep the dimension n constant. It can be actually the opposite. Sometimes when you work within one omic, you increase the dimension n actually. If you, for example, working with RNA sequencing, and you happen to combine data sets from different labs or different experiments, then your P dimension will be constant, but N dimension will be increasing. So it's another type of integration, which is called vertical integration. But in this lecture, I will be not talking about vertical integration, but will be concentrating on this horizontal integration. Because vertical integration, you can think about it as a batch effect correction. So it's quite an old problem, and there is a lot of development in this area. So it's a little bit maybe less challenging, but what is sort of not straightforward to understand what exactly we are doing is when we put different data types together, when we put protein abundance, gene expression, and maybe some genomics data together measured for the same biological samples. So I'll be talking about this horizontal integration when you put different data types together measured for the same biological samples. And sometimes you can have pretty much squared matrices. For example, for metabolomics and proteomics, we typically have N and P comparable with each other. So they are on the same order of magnitude. N can be a little bit larger or smaller, but they are typically comparable. And so I call, these particular data types, metabolomics and proteomics, they're pretty straightforward to use for integration, data integration. So I call them manageable. So there are no sort of statistical issues integrating these data types. While if you move into transcriptomics, here you have a situation when P dimension can be much greater than N. So this P dimension is elongated a little bit. For example, if you're working with human genome, your P dimension is 20,000, approximately 20,000 genes, protein coding genes, and N, typically how much do we have? Maybe 100, maybe 1,000 samples if it's bulk RNA sequencing. In contrast, in single cell transcriptomics, you actually have N very large. So basically in single cell transcriptomics, this is less of an issue, or this high dimensionality of the data is less of an issue. So what I'm talking about now is that when we sort of increase in this P dimension, we are going to high dimensional data. And this can be a big challenge for data integration. So two other types, typical types of biological data, genomics and metallomics methylation values. It has a very, so it's basically, it's a very much elongated in the P dimension. So these data types where P is much greater than N, those are particularly challenging for data integration, for omics integration. And I call them impossible, or it's possible still, but it's very challenging. So why I'm worried about this high dimensionality of the data, you can ask. So why I'm worried that making this horizontal data integration, they're creating high and ultra high dimensional data. Well, it has something to do with a problem called the curse of dimensionality. So in data analysis, there is a general and long-standing issue. It's called the curse of dimensionality. So what's that? So as I said, typically working with matrices as a typical representation of the data, you have P and N dimension. And depending on the ratio between N and P, whether P is greater than N or N is greater than P, you can approximately choose between those three types of statistical analysis. So if you have P much greater than N, you have very little data. Your N is small, and you better take this into account. You should use some regularizations which are naturally implemented within Bayesian type of analysis. So you probably should be doing Bayesian statistics if you have very little data. If you have enough data, when P and N are comparable, you can proceed with traditional frequency statistics. This is Roland Fisher, the founder of frequency statistics. The statistics with p-values, with hypothesis testing, with power analysis, something what we consider traditional statistics. If you have a lot of data when N is much greater than P, you can still do frequency statistics or Bayesian statistics, but you can actually do better than this. You can go beyond linear analysis, because frequency statistics essentially is based on linear algebra. It's a linear type of analysis, but when you have a lot of data, you can afford going beyond linearities and doing machine learning and even deep learning type of analysis. So usually medical and biomedical and biological data is so if you look at the dimensions of typical biological data set, it will be probably here. It will be probably P will be much greater than N. As I said, for a typical RNA sequencing transcriptomics data type, you have 20,000 genes, and N just a few hundred or maybe 1,000 if it's not single cell transcriptomics. So what would be a danger of doing sort of frequency statistics, for example, in this limit, if you have very little data, but you still try to do hypothesis testing, for example? Well, one danger is basically explained here. So if you're doing a simple linear regression analysis, if you have a dependent variable Y, maybe a phenotype, and X is a genotype, for example, if you run this association study type of analysis, so the solution of this linear regression problem can be expressed in closed form as follows. And here you have an inverse variance-covariance matrix. And it can be shown that if you compute this variance-covariance matrix, it diverges in this limit, P much greater than N. So so basically doing simple analysis, frequentist type of analysis, such as linear regression, without taking into account high dimensionality, high dimensional nature of biological data, you might get into singularities, and your R or Python can still give you a formal answer, but there is a danger, there is a risk that this answer is inflated, and you never know how close to this singularity point you are actually. Another example of sort of misusing assumptions of frequentist statistics working in the limit p much greater than n, high dimensional limit, is the very famous biased maximum likelihood variance estimator problem. So basically, when you do hypothesis testing, you always need to compute variance or standard deviation in your data. It can be shown sort of that if you compute variance within maximum likelihood framework, which is dominating in frequentist statistics. So your estimated variance on the left-hand side of this equation is expressed through true variance, sigma squared, on the right-hand side of this equation, through this coefficient n minus p divided by n. And here you have an explicit interplay between n and p. So basically, you can think that if p is increasing, if p is growing, your estimated variance is becoming further and further away from the true variance. So you're basically estimated. You cannot correctly, robustly estimate variance in your data doing frequentist statistics in the high dimensional limit. So these problems, they happen when you have high dimensional data, and biological data is high dimensional. So when you do data integration, you further increase dimensionality. So that's one of the challenge of data integration, how to avoid the curse of dimensionality, how to combat the curse of dimensionality. Just a very brief example of the curse of dimensionality, how it's manifested. If you try to compute distributions of pairwise distances between data points in 2 dimensions, 32 dimensions, and 512 dimensions, and plot this histogram of pairwise distances between data points for different dimensionality of your data, you can notice 2 things. First, the data distribution is shifting towards the higher average distances between data points. So meaning that points are becoming further and further apart from each other in high dimensions. Second thing you can notice is that the high dimensional data has a more narrow pairwise distance distribution, meaning that the data points are becoming more and more similar to each other. And also, if you just for a random point, you compute a distance to its first neighbor, to its middle neighbor, and furthest neighbor as a function of dimensionality, you can notice that in high dimensional limit here, the distances between nearest neighbor and furthest neighbor, the difference between them disappears. So the nearest neighbor and furthest neighbor, they become basically indistinguishable. And this is a problem, because so it means that in high dimensional space, there is no difference between cases and controls. You can't really cluster them. So this difference disappears or gets masked in a high dimensional limit. So this is just an illustration of that in high dimensional data is special. And when we do data integration, again, we are going towards high dimensional data, which needs to be treated carefully and properly taken into account. So happily, we have another data type, which is a single cell transcriptomics data. And it's becoming almost a routine to sequence hundreds of thousands or even millions of cells, meaning that your n dimension is actually hundreds of thousands or millions. So you actually you're sitting in the opposite limit when you do not have that much a problem of a curse of dimensionality, but you better so you should not be worried about overfitting your data, but you rather watch out for underfitting your data. And in this case, it's a good limit to try non-linear approaches of data analysis, such as machine learning and deep learning. I've seen that there was a raised hand. Maybe we can take a question.
Yes, hello. The question was, where do the data that you've shown in the distribution before come from? So is this specific biological data, or is it something that is valuable more in general? Thank you.
Yeah, so thank you for the question. So what you see here on the screen, this is simulated data. So you can simulate high dimensional data. However, so if you're interested, I can sort of give you examples of real data types. When you sort of take RNA sequencing data and try to decrease dimensionality of the data and see and measure pairwise distances. So basically, I have this sort of analysis done that shows basically the same type of behavior as on simulated data. So basically, despite this is simulated data here, my strong belief is that the qualitative behavior of these distributions of pairwise distances will be the same for, well, I tested it for RNA sequencing, but I believe it's valid for many types of biological data types.
So is this data generated randomly, or are you following some rule for the generation?
It was multivariate Gaussian distribution, yes. So the data was drawn from multivariate Gaussian distribution.
Okay, perfect. Thank you. Very interesting.
So now I hope I delivered my message that the curse of dimensionality is one of the major challenges in omics integration. So everything else what we'll be discussing today is kind of it's all coupled and connected with this constant battle how to overcome the curse of dimensionality. But having said that, now I want to formally introduce what I mean by machine learning type of data integration and how I evaluate that the integration worked successfully. So basically, if you search in literature, omics integration and data integration, there are plenty of methods available. Basically, you can go to the details of each method and learn how it works. So here I just mention a few of them. I think more important is to understand that all these methods they essentially do similar thing, and this is illustrated here. So the idea of any machine learning type of data integration is, or maybe even data integration in general, is to see something new in your data, something that was hidden in individual omics. Once you put different data types together, you start seeing new patterns that were not visible before integration. And to illustrate this, so here on this plot, one point is one sample. And these samples belong to three classes, class 1, 2, and 3, black, red, and green points. Imagine that you have two omics, maybe RNA sequencing and methylation data, omic 1 and omic 2. Now you can create a latent variable from omic 1. It can be, for example, principal component 1. So you take RNA sequencing, you you do principal component analysis, and you take PC1 and plot it as x-axis. And then you do principal component analysis on methylation data and plot it as y variable here. And you take PC1 from omic2 from methylation data and plot it as y-axis. So it turns out that if there is a covariance between those two data types, if there is a correlation, if those two data types are related, you can discriminate between the three classes much more clear. So, for example, if you are using just one data type, just RNA sequencing, imagine that you can project those points on the x-axis, and you will see that the points, the black, red, and green points, they're mixed, so they're not separated. You don't see a clear separation between the three classes by using just one omic. And the same as well that if you project the data points on the y-axis, you can't really discriminate between classes using just single omic. Although if you, however, if you put those two omics together, if you put the latent variables constructed out of the two omics, if you plot these two latent variables constructed out of the two omics against each other, you will see that if there is a relation between the omics, the black, red, and green points can be linearly separated. You start seeing separation of the classes when you combine omics. That's a beauty of data integration, that you see patterns that were invisible when you're working with individual omics. A good tool and a good good method that I recommend everyone is to visualize if there is a connection actually between omics, genome-wide connection, is this Morpho tool that delivers this beautiful heat map. And what it does, sort of, we will cover Morpho a little bit later, but briefly, it sort of decomposes, it does a factor analysis type of, and it decomposes your data into some latent factors, splits your data into LF1, 2, and 3. And from this heat map, we can see that single-cell ATAC-seq data was covariant with single-cell RNA-seq data in latent factor 1, while single-cell bisulfite-seq data was covariant or correlated with single-cell RNA-seq in the latent factor 2. So basically, indeed, by sort of doing this factor analysis with Morpho, we can quickly check whether there is a relation between the two omics, and then we can proceed with integrative analysis. Then it makes sense to do omics integration. We can expect to see some hidden patterns. So yes, I can see there was one more hand raised.
Yeah, very quickly, I'm not very familiar with the concept of latent variable. Can we assume that latent factor 1, 2, and 3, they correspond to principal components 1, 2, and 3, so they represent decreasingly amount of variance?
Yes, absolutely. So if you're talking about principal components, yes. So it's basically decomposing your data with respect to variance.
So can we say that in the heat map, the purple heat map, the higher lines, so LF3 and LF2, are a little less important than the first one, LF1?
Yes, that would be my interpretation, yes. But it should be sort of, I can't answer this question like for sure, because you need to sort of understand better Morpho. But from my understanding, yes, I would say so.
Okay, thank you.
Yes, so now once we defined what we mean by integration, but by data integration. Let's talk about how we evaluate whether integration was successful. And then here we can use some experience from data science. We can borrow some principles from data science. When people working using data in the internet, they often work with texts, images, and sound, for example, and maybe you can do some sort of predictions about what kind of products, what kind of what kind of books people like, or what kind of images they would like to watch, what kind of movies. So you can create people often. I mean, in the internet, we have recommender systems, and you can sort of predict something about user interests using individual data types, such as text, image, and sound, and achieve some accuracy, maybe 78, 83, and 75% of accuracy. So the numbers are made up, just an illustration. So you want to predict something about the users. For example, their gender, whether it's a male or female, based on the data that they're using. And so the idea of data integration in data science is that if you put all the three data types, text, image, and sound together, we would expect to increase the accuracy of prediction, something about the users. So basically, we by combining different data types, we see we can expect a boost in prediction, and this can be useful to create a more accurate recommender systems. So we can use in life sciences, in computational biology, we can use similar principles and use prediction and a boost in prediction as a criterion of successful integration. So in here, so that's an essential concept. So prediction is very essential for machine learning. So any machine learning delivers you a model that predicts something, which is a little bit counterintuitive for people with pure biological background. They usually may be interested in biological mechanisms, and it's not always clear why we would like to use prediction, why we would like to predict. And people doing traditional mathematical statistics, they often, so that's actually a difference between traditional mathematical statistics and machine learning. In traditional statistics, we often, or in traditional computational biology, people often search for biomarkers. They want to find genes or proteins that are coupled to maybe disease. They want to find biomarkers. And they do this so-called univariate selection when they test gene by gene or marker by marker for its relevance to a phenotype, such as a disease. For example, like in genome-wide association studies. The consequence of this type of approach, when we don't really optimize prediction, is sometimes you can get into the problem, which is called the case of the missing heritability. That your biomarkers do not really predict the disease. In contrast, machine learning, when you do machine learning type of analysis, you always optimize a prediction, and you're not necessarily interested in biomarkers. While in fact, biomarkers can be delivered as a consequence, as a byproduct of your machine learning type of analysis. But you are optimizing predictions from the very beginning. That's why you would have a less of a problem with a missing heritability if you started with machine learning type of analysis with optimizing prediction from the beginning. Because the case of the missing heritability, the problem of the missing heritability, it's basically a lack of prediction on an independent test data set, which it might happen if you sort of search for candidates and not necessarily try to select candidates that predict. So I hope that's a clear conceptual difference between traditional statistics and machine learning. So in our biological sciences, the way we sort of similar, if you borrow the same idea from data science, if you use prediction as a criterion of successful integration, we can reformulate it as follows: that maybe if you have methylation data, gene expression data, and phenotypic data measured on the same biological samples, you can see that they all have very different data distributions, so it's not straightforward to combine them. However, maybe we can use individual data types in order to discriminate between healthy and sick individuals, and we can achieve good accuracies maybe of classification, healthy versus sick. However, the idea is that of data integration is that if we combine those different data types, the goal would be to see an increase in prediction, increase in accuracy of classification, healthy versus sick. And how to do it? There are different ways. Here I briefly cover sort of three directions of different methods, integrative methods that have sort of machine learning philosophy. So you can convert your data to a common space, to some non-parametric space where they forget about their technological past. So neural networks, similarity network fusion, or UMAP, this is what essentially they do. You transform, you do some weird transformation with your data, and you keep only pairwise relations between individuals without sort of accounting explicitly for the biological nature of the data type. Second approach is that you can explicitly model this distribution. So if you can't convert the data to a sort of common data distribution, you can try to explicitly model this distribution. So Morpho and Bayesian approaches, those are explicitly modeling this distribution through joint probability distribution. So if you remember, the Bayes rule is basically it's a joint probability distribution from different data types. So it's ideally suited for combining data from different data distributions. So Bayes rule is ideally suited for this. And the third direction is extract common variation from different data types. So you basically you try to decompose your data into or split your data variation in extract common axes, common sources of variation from the data. You don't really model your data distribution explicitly in this third direction, but it's a very popular type of data integration analysis: PLS, partial least square regression, CCA, canonical correlation analysis, or factor analysis. This is what essentially these methods are doing. And here I will stop this first lecture. Machine learning view of omics integration. So I hope I delivered three main concepts: that the curse of dimensionality is a major challenge. Omics integration can be viewed as a way to discover new hidden patterns that were not visible in individual omics. And the third message is prediction or increase in prediction after integration can be a criterion of successful omics integration. So I'll be happy to take questions, or maybe continue. Rui, what would you say? Continue?
Yes, I think we continue for now. And if there are any questions, people are very welcome to ask them on HackMD.
All right, I see that I'm behind the schedule. So I will try to maybe then I'll try to concentrate on the major like the main slides. Then in order to in order to cover basically most important things. Yes, so I think having said those things about definition, how we define machine learning omics integration and prediction as a criterion of successful omics integration, we are basically ready to formulate some sort of protocol of omics integration. And the first question we can ask, or very often people ask, what method of integration I should select for my data. And here I think it's useful to keep in mind this kind of table and understand, realize that data integration methods can be sort of divided into linear and non-linear, and also supervised and unsupervised types of analysis. For example, if you have a lot of data, maybe you should consider doing non-linear type of analysis. While if you have just a few samples, you cannot afford to do non-linear analysis. You should better stick to linear regression or p-less or LAS or this type of linear analysis. Simply because so the rule of thumb is the more data you have, the more you should consider trying non-linear types of analysis. The less data you have, you better do linear type of analysis. And then supervised and unsupervised, the choice of supervised or unsupervised analysis depends on if you have a very clear phenotype of interest in your biological data. So for example, a disease. So if you're collecting, if you're doing a biomedical research and you have samples from healthy and sick individuals, so the disease status is a very clear phenotype of interest. You can center all your analysis using this phenotype of interest. So to give you an example, so for example, if I have 110 samples, and those samples have maybe gene expression and genomics data and protein abundance data. So we are talking about high dimensional data with only 100 samples. It's a good idea to do linear type of analysis. You can't really afford going beyond linearities. And then if you have a type 2 diabetes, for example, if your data were collected for diabetics and non-diabetics, so you have a clear phenotype of interest. You can do supervised analysis. Otherwise, if you're not sure about what is in your data, you better sort of you can do unsupervised analysis. And here I mention a few methods that sort of represent linear supervised. It's p-less, what we will be covering basically here. Partial least square regression, it's a linear and supervised type of analysis, while maybe UMAP, it's a non-linear and unsupervised type of analysis. So basically the choice of integrative methods, in order to answer the question, what kind of integrative method I should choose, you should think about the amount of your data and whether you have a clear phenotype of interest. So for example, in this example with 110 samples and T2D samples, you can think, you can create this kind of simple integrative loop. So if you want to do a machine learning type of integration, so you should, from the very beginning, you should split your data into train and test data set, because the test data set will be used at the very end to evaluate your model, to evaluate whether your method worked successfully, whether you actually see the boost, the increase in prediction. On the trained data set, you very often need to perform feature pre-selection to reduce dimensionality of your space. So it would be very naive to put high dimensional data together without trying to somehow reduce this problem of the curse of dimensionality. And this is what this feature pre-selection is supposed to do. And you can do unsupervised feature pre-selection or supervised. So with unsupervised feature pre-selection, you can just, if you don't have a clear phenotype of interest, you can just look at the variation between samples and remove features that are non-varying between samples. Those features are probably non-informative. If you have a clear phenotype of interest, you can just perform some sort of association with your phenotype of each feature. You can sort of do regression of each feature against your phenotype of interest and select most relevant features. So next, once you've done feature pre-selection, you can combine actually them as in the integrative omics using your favorite method, the method that you can pick from this table. And once you performed omics integration, then the results of your model, so the performance of your model can be evaluated on the test data set. And hopefully and ideally, you should see some increase in prediction, T2D, diabetics versus non-diabetics. This is what in this example I'm trying to classify. This increase in prediction, the better, more accurate prediction should be a consequence of data integration. So it should ideally outperform similar type of prediction based on individual omics. So basically here in a few lines, I sort of summarize this protocol that I use and I sort of suggest that you might consider to use for your projects. So first we check whether there is a relation between samples, and here Morpho is a great tool. Then we choose the integrative model based on the amount of the data. It can be linear and supervised or unsupervised or non-linear method. We do feature pre-selection on the trained data set. We integrate our data on the trained data set, and we evaluate our model on the test data set and compare the performance of the integrative model against the corresponding performance of models on individual omics. And ideally, you should see some synergistic effects. I was talking a lot about feature pre-selection as a way to overcome the curse of dimensionality, to make your data less high dimensional. And there are basically two types of feature selection, univariate feature selection and multivariate feature selection. In the practical exercise after this session, I'm sort of covering this example of gene expression data, GTX gene expression data from human skeletal muscles. I'm trying to sort of find genes that discriminate the most, whose expression discriminate the most between males and females. And if you just perform principal component analysis on the approximately 150 samples from GTX, gene expression on human skeletal muscles, you don't see that males and females are separated, so they don't see a clear separation between males and females. However, you can test one gene at a time against your phenotype of interest, which is female and male. So your gender is your phenotype of interest in this particular toy example. And you can rank all your genes based on how well each of the genes is correlated with gender. You can sort of remove genes that are not correlated with your phenotype of interest. That would be a way to pre-select features. But here you are testing one gene at a time. This is why it's called univariate feature selection. And there is a lot of evidence that basically this univariate type of analysis has a lower predictive power, predictive capacity compared to multivariate type of analysis, such as lasso. Lasso, here, when you're doing lasso, when you're doing feature pre-selection with lasso, you basically you're trying to prioritize features such as genes. So here you have a toy example of two genes, X1 and X2. Those are two genes, and Y can be your male or female, the gender phenotype. This is a linear regression problem, and you typically solve this linear regression problem by minimizing your residuals, your error of your model. So you use your data and you subtract your data from your model, and this is the residuals, and you try to minimize this error of your model. This is how a typical linear regression works through ordinary least square algorithm. But you can do it better with lasso when you are adding, so you're basically using the same OLS, ordinary least square regression principle, but you're adding some penalty. And this penalty is supposed to select only most relevant, most informative features. And this lambda coefficient is called Lagrange multiplier. It's a hyperparameter of lasso, and it can be found through cross-validation. So what you see here is actually an error of the model plotted as a function of lambda parameter. So you can always do, by doing cross-validation, you can find a value of lambda where your error of model prediction reaches its minimum. And this will be the optimal lambda parameter. And cross-validation, what it means essentially, you split your data sets into a few parts, if you're talking about k-fold cross-validation. And you train your model on so if you split it into 5 parts and you train your model on 4 parts and you evaluate your model on the last piece. And this model will always have an error. So by summing up errors from different k-fold cross-validation rounds, so you can actually, this is exactly what you see here on the y-axis. So y-axis is just a summed error of different cross-validation rounds. So so I hope that's a clear difference between univariate and multivariate feature selection. That here, when you're using lasso, you're actually putting all your genes together. So X1 and X2, you're not checking, you're not testing them one at a time. So you're putting all your features, X1, X2, etc., together, and you use some priority rule, which is this penalty, which is implemented within lasso. And this priority rule will pre-select the most relevant features. So I see that I'm running out of time. Maybe I can briefly sort of go through the few most important slides of supervised integration after the break, because unsupervised part is not as large, and I hope I will I suggest to finish this presentation after the break.
All right, so let's take a 5 minutes break and we will come back soon. Nikolay, we can see your screen, but we don't hear you or see you. Here we go.
Yes, apologies. Yes, so thank you, welcome back. So I briefly explained here how the difference between univariate and multivariate feature selection. So again, univariate feature selection, testing each feature one at a time and selecting most relevant features. Multivariate feature selection, supervised feature selection is testing all features together and selecting most relevant ones through the penalty, through some factor that counts for that sorts out noisy features. So basically there are three types of penalized regression models. Those are lasso, rich, and elastic net. You probably heard about them or using them. Sometimes there is some confusion about what's the difference between them and which of them to select, which one to use for my type of analysis. So the difference is in this penalty term. So basically they're using slightly different penalty terms. And to make long story short is that you can say that lasso is a more conservative out of all three of them. Lasso is most conservative and Reach is most permissive out of them. However, for noisy data types, for biological data types, I would recommend to use Lasso as a more conservative penalized model that selects only the most reliable features. And moreover, from my sort of extensive testing, you can also clearly see that Lasso usually provides a better, so features selected by Lasso usually have a higher predictive capacity, which is sort of seen here on this ROC curve, where Lasso and elastic net ROC curves, they're clearly above the Reach ROC curve, and this was done for this gene expression data type, and the phenotype was gender. So at least for this toy data set, it's clearly you can clearly see that the Lasso is has a higher predictive capacity compared to Reach. So next, there is another model of linear feature selection, which is a partial least square regression. So basically, you should remember three major models for supervised linear feature pre-selection. Those are penalized models such as Lasso, Reach, and elastic net, and PLS, partial least square regression, and LDA, linear discriminant analysis. So here, I briefly explained the idea of partial least square regression. You will be using it during the hands-on session. You'll be doing omics integration through mix omics package, which is based on PLS. So it's good to understand what PLS is doing. So basically, you've seen that males and females are not clearly separable on this PCA plot. However, if you select proper features with PLS, you suddenly can see clear separation between males and females. So basically, you can think about PLS as a method that selects features, select genes in this case, that separate two classes the best, the most. Brief idea, and you can always sort of understand what features drive this separation. So you can use this loadings, so-called loadings, and you will see it in the exercises that you can rank all the genes by their contribution to this separation. So you can select most informative genes, the genes that discriminate between males and females the most. So the third type of linear feature selection model is the linear discriminant analysis. So all these three types, Lasso, PLS, and LDA, they are conceptually similar. So they all are linear and supervised. So essentially, it doesn't matter much which one of them to use for your particular analysis. They, to my experience, all three of them should deliver similar type of ranking of most informative features. So what LDA does, sorry, it's not gene expression data set in this case, but it's another benchmark data set. So again, it tries, LDA tries to maximize variance between clusters and minimize variance within clusters. So in this way, you can think that by selecting features that maximize variance between clusters, you can sort of split and make these clusters more distinct. If you try to maximize the variance between clusters and minimize the variance within cluster. So in this sense, if you're familiar with ANOVA or t-test, that those methods are also doing conceptually this type of thing of maximizing between variance between clusters variance. So I'm moving towards the major method that you are going to cover in the hands-on session, which is a Diablo integrative method, which is based on PLS. That's why I explained PLS, briefly explained PLS first. So Diablo is developed by Kim-Ann Lee Kao from Melbourne and her team. It's a very elegant method, which I highly recommend you. And all tools in the multi-omics package, they are very statistically robust, and I use them a lot. So the idea behind Diablo is, as I said, it's a PLS inspired, and basically you can think about integration of two data sets, X and Y, as maximizing covariance between those two matrices. So if you have two data matrices, X and Y, so you can try to maximize covariance between them and optimize parameters of the model by maximizing covariance between X and Y. And then you can sort of split this maximized covariance in the independent components in order to interpret the results. So this is what essentially Diablo is doing. And in the methods section of Diablo paper, they basically write this equation 1, which is sort of, it looks scary, but if you sort of think about this equation, you can see that if you have two data types, two matrices, xi and xj. So what Diablo essentially does, so this x times a, it's basically a principal component of data type 1. And here is a principal component of data type 2. So what you are trying to do, you are trying to maximize correlation between leading principal components from two separate omics. And then this is how you do data integration in Diablo. Diablo has very good visualization. So you can, so you will be playing in the exercises, you will be playing with different visualization in Diablo. So basically, in the data set, in the benchmark data set, you'll be using, there are 4 omics: gene expression, mutations, methylation, and drug response, measured on some cancer cells. And what Diablo, sort of the major result of data integration through Diablo, it's this arrow, so-called arrow plot, when you clearly can see that after the integration, the two classes, the orange and blue, they become very distinct. And you can also sort of decompose. So this is a result. This is a consensus plot. This is a plot you obtain after putting all 4 omics together. But if you want to split this consensus plot into sort of parts for each omic. You can also see that gene expression maybe contributes the most to this consensus separation. You can also sort of look inside and understand how each omic contributes to this consensus. And once you've done the integration through Diablo, you can test your model, the performance of your model on a test data set that you kept separated from the very beginning. So basically, this plot, the error plot, the consensus plot, was produced on the trained data. And what this PLS classifier learns, it learns to draw a boundary between orange and blue samples. So this is how you can visualize it. So if there are two classes, so the classifier sort of converts your data in some space where two classes become linearly separable. And then you sort of can add test samples. So the field points here, the field circles here, those are test data points that were not used to train the classifier. But you can sort of add them and see that the classifier actually can, the classifier can predict their class very well. So usually the red field circles, they fall on the right-hand side from this boundary, decision boundary between the two classes. But sometimes the classifier can misclassify certain data points, such as this black data point. It falls on the right-hand side from the decision boundary. So the integration and the classification was not ideal. So there is some error. So I skip maybe some other types of visualization in Diablo. You can sort of look at them in the hands-on session in the notebook. But I briefly mention a few other types of supervised data integration, how you can do data integration in, for example, a non-linear way. So Diablo and PLS, those are linear supervised data integration types. While using, for example, neural network, you can, it's a pretty straightforward way of data integration where you can play and you can add non-linearity. You can use non-linear types of analysis. And sort of the way you integrate your data through a neural network is that you simply, you convert each of your omic into some hidden space, hidden layer, and you merge your omics converted into hidden layers. You can merge them together. And since a neural network has a rule of updating the weights, updating the parameters of the model, which is called backpropagation. So it means that the weights, the parameters of the model will be updated in the context of each other for all omics simultaneously. So basically, the parameters for omic 1 will be updated, taking into account parameters from omic 3, for example. So you update, you do updating of your model parameters by keeping all your omics together. So this is where the integration comes. So this is a pretty straightforward way of data integration. So you do here integration by merging your omics in the hidden layer, and then on the output layer you do prediction. So similar to PLS, you might have a phenotype of interest such as gender, for example, and you predict the gender on the output layer. So another interesting type of supervised omics integration is Bayesian networks. It's a sort of maybe more biology-friendly way of omics integration when you explicitly can manipulate connections between different nodes. So that there are interesting packages developed for doing sort of Bayesian network analysis. But still they need some feature pre-selection before putting your data into a Bayesian network. You still need to reduce dimensionality of your data, such as gene expression metrics, by using some sort of clustering. Dimensionality reduction should be done prior to applying Bayesian network. Because Bayesian network is not very scalable framework, so it unfortunately cannot scale for a large amount of features. So you have to pre-select features before using Bayesian network. So I'm not sort of going into details, but just mentioning that there are interesting ways of integration, supervised integration. And there is, I also just mentioned it here, there is a there is a methodology. So, and I refer to this paper, which is data integration through random forest. And the idea is that random forest essentially sort of categorize your data, splits or dichotomize your data, splits into categories. And that's why this is how you overcome this issue of data coming from different distributions, because after this categorization in the random forest tree, you end up with data more or less coming from the same distribution. This is the idea behind data integration through random forest. So, and here I stop the supervised omics integration lecture, and I'll move to unsupervised integration part in a second. Yes, so in unsupervised omics integration, we don't have a clear phenotype of interest, and basically very often the problem is formulated as follows: find something in my data. So I have maybe gene expression, protein abundances, and maybe genomics data from the same biological individuals. And I think I explored them well enough, but maybe if I do some unsupervised type of analysis, I can discover something new, something that I never thought about. And in this sense, it can be sort of viewed as a little bit like a fishing expedition. So an analysis without clear goals. MOFA, MOFA, what you're going to sort of test in the hands-on session is a very elegant, very interesting tool, hypothesis-free tool. So when you don't have a clear phenotype of interest, and the idea of MOFA is essentially what MOFA does, it decomposes your data, your variation in your data into common variation, shared variation between different omics, and technology-specific variation. And once you sort of extracted common variation, you can split it into orthogonal components in order to sort of understand, interpret, and visualize your data. So the idea behind MOFA is factor analysis, which essentially means extracting common variation and factorizing it, extracting common variation from your data and splitting it into orthogonal components. This is the idea in a few words. And it has, so MOFA has, again, wonderful visualization, and it actually can do data imputation, for example, which is a big problem in data science, that you often have missing data, and removing samples with the missing data is not really a good idea, because you can introduce some biases. So what MOFA can do is, because it's a Bayesian framework, you can, if there are some data missing in omic 1, you can still include this data into your analysis, because the other omics, they will help to predict, to impute the missing values in omic 1. Yes, so in the MOFA paper, they test this imputation, and this is quite elegant, that you can sort of see that due to integration, you can actually fill the gaps in each individual omic. Yes, so in the hands-on session, you're going to apply MOFA to this SNMT dataset, which is it's a single cell multi-omics dataset, and it consists of RNA sequencing, bisulfite sequencing, and single cell attack sequencing data measured on the same cells. So it's not really a high throughput data. It's not high throughput method. SNMT is not really high throughput because yeah, there are some technological limitations. So it's only done for 200 cells, but still it's interesting because all the three data types, they're measured on the same biological cells. And if you visualize the data distributions from single cell RNA-seq, bisulfite-seq, and attack-seq, those data distributions, they look very, very different. So again, meaning that just merging, just concatenating data sets, it's not really a good idea, not really an option, because you can't properly model the resulting data distribution. You have to have some idea about what kind of data distribution you'll get at the end. And the way MOFA puts these different data types together is through this Bayesian rule. So basically, when you're working, so it has this Bayesian internal framework, and the Bayesian rule. It's in the numerator of the Bayesian rule. There is basically a product of probability distributions from different sources. Sometimes they call the prior is called user experience. So, but it's essentially it's another sort of data type. So you can use Bayes rule and add as many factors in the numerator of the Bayes rule as you want. So essentially what you get a joint probability distribution. So your resulting posterior probability distribution, it's a joint probability distribution from all your omics, multiplied and combined together in the numerator of the Bayes rule. Hope this is clear. So MOFA delivers this beautiful visualization of importance and contribution of each data type to the resulting variation in your data. So this is a heat map that I already mentioned, where all the contributions are sort of visualized or colored. As I said, when you do MOFA, you basically you extract common variation from the data and split it into orthogonal factors, which are called latent factors. And this is what is plotted here. on the y-axis of this heat map, those are the latent factors, LF1, LF2, and LF3. Those sort of they come from splitting the common variance into orthogonal components. And then you can understand what omics contribute to each latent factor. And since it looks very purple here for single cell ATAC-seq and RNA-seq in the first latent factor, you can expect that there is something in common between those two omics, RNA-seq and ATAC-seq, and you can interpret and find the links between those two omics by looking at the loadings of latent factor 1. And similarly, bisulfite-seq and ATAC-seq, they are combined, or they are co-occurring within latent factor 2, meaning that looking at the loadings of latent factor 2, you can find links and connections between those two omics, between single-cell RNA-seq and single-cell bisulfite-seq. Yes, and this is how you can sort of visualize the loadings. So looking at each loading, you can understand also individual features that contribute to each loading. And here there will be so this is a way to understand how features are connected across omics, how genes, what genes from gene expression correspond to what methylation markers from single cell bisulfite-seq data, for example. And as a result of MOFA integration, you also get a consensus plot. So those cells, 200 cells approximately, now they're visualized in a common space. So this is not just RNA, it's not based on just RNA sequence, bisulfite-seq. This is this plot, what you see is a result of integration. It's a result of combined omics, combined single-cell RNA-seq, bisulfite-seq, and ATAC-seq data. And sometimes this can bring a new resolution. Sometimes you can see patterns. Sometimes you can discover cell clustering that was not visible from, for example, single-cell RNA sequencing alone. So MOFA is also originally MOFA was developed for bulk sequencing, but the second version of MOFA, MOFA 2, was also extended to single cell data. And in their paper, they the authors of MOFA, they are emphasizing that by combining RNA sequencing and ATAC-seq data, you can produce this sort of consensus plot similar to what I've showed here. And sometimes this can improve the resolution and bring you more distinct clusters. In single cell analysis, you are often you want to discover new cell populations. And the tricky question is whether so if you add epigenetics to transcriptomics and you suddenly see a new cell cluster, does it mean that you discovered a new cell type or not? That's a tricky question. So basically, for a very long time, people were learning new cell types by looking at transcriptomics alone. Single cell RNA sequencing was dominating the single cell area. But then a realization came that if you add another levels of information, such as epigenetics, you might see that uniform clusters, they suddenly split, meaning that maybe This means that this cluster was not uniform, and there are also subtypes, cell subtypes that become visible after you add epigenetics information. Another beautiful and impressive thing about MOFA2 applied to single cells is that you can actually look at these clusters and understand whether they are driven by factor by latent factor 1, 2, or 3, etc. So, for example, here on this plot, so maybe the coloring of the cells is by factor, and meaning that this cluster is driven by factor 4, for example, and you can immediately see that this cluster is probably, if it's a new cell type, then it has something to do with ataxic. So it's not really driven by RNA sequencing. So you can you can see what omic contributes the most to each cluster. This is elegant. Other types of unsupervised integrative analysis. So now I will also mention briefly a few other types of unsupervised analysis beyond MOFA. So MOFA is a linear type of unsupervised analysis, but you can sort of use, for example, a very old concept of clustering of clusters. This is sort of how unsupervised omics integration traditionally started some time ago, when people tried to establish links between samples within each class, within each omic, I'm sorry. And once these samples were clustered within each omic, then those clustering results, they were overlapped. This is where integration came. So basically, the idea is very simple. You cluster your samples within individual omics and then overlap the results. This is what is clustering of clusters about. A famous model, or very one of the first models using this concept of clustering of clusters was similarity network fusion. So the idea is the same, that you if you have 3 omics, you can try to establish links between samples using individual omics, and then use some sort of rule or algorithm for overlapping the links. So imagine that so you have 3 clusters in each omic, and then you would like to establish some sort of protocol or rule on what kind of links between samples you would like to keep when you overlap the clustering results across omics. And at least in this similarity network fusion Nature method paper, they demonstrated that you can, by using this clustering of clusters approach, you can remove technological noise, which was technology specific. So maybe here there was no sharp clustering, and here there was no sharp clustering. But once you overlapped those 2 omics, your overlapped clustering results, you get very sharp, very distant clusters, because you sort of concentrated on the common links, common across omics links between samples, and removed the noisy links. And here we come to another direction in unsupervised omics integration, which is a graph-based omics integration, which Rui will be talking about tomorrow. And the idea is, again, the idea is simple. We build graphs for each individual omics. For example, using this SNMT dataset above, consisting of single-cell RNA sequencing, bisulfite-seq, and ATAC-seq. We can try to create a graph to visualize samples, to visualize cells by building a graph in each individual omic. And then we a possible way to perform integration once you constructed individual graphs is to keep edges. So a graph consists of nodes and edges, and you can try to keep edges that are consistently present across 3 data types. So if an edge, for example, here highlighted by red, if an edge between 2 cells was present here and an edge was established here between the same cells in single-cell bisulfite-seq data, and the edge was established here between the same 2 cells using ATAC-seq data, then this edge should be kept in the consensus graph. This is a simple graph intersection rule. When you have sort of looking at this image, you can see that the resulting graph, the intersection graph, it keeps only edges that were present in both G and H graphs. So this is what is demonstrated here. So you can visualize all 3 omics by keeping consistent edges. And this is basically what is implemented in UMAP. So UMAP is very fast and very elegant and very simple way of unsupervised omics integration. Maybe if you are sort of puzzled about MOFA and don't understand technical details, and maybe it takes a lot of time to run MOFA, which can be the case, but you can maybe choose UMAP. That is basically to do data integration with UMAP. It's a few lines of code only, again, what UMAP is doing is basically it overlaps graphs and keeps consistent edges, but it's done sort of internally in UMAP, and you don't really you don't really have to be concerned about sort of technical details. You don't need to construct those graphs by yourself. So it's if you perform unsupervised integration within UMAP, it's already there, and it's just 1 line of code. And the results of omics integration through UMAP can be visualized as follows. So if you have a UMAP plot on single-cell RNA-seq data, and you have a UMAP plot on single-cell proteomics data, you can see that they're a little bit different. So they don't tell you same stories. Some clusters are unique to single-cell proteomics, and they're not present or not very obvious in single-cell RNA sequencing data. But when you put those 2 omics together, it can help you to better see certain cell types, such as so on the UMAP plot on the bottom of the screen. This is a consensus plot. This is a result of integration between transcriptomics and protein abundance level of information from a technology called CiteSeq. And here, for example, this purple cluster becomes more visible. So if you were using single-cell RNA-seq data alone and run clustering on this image, you would have problems identifying the purple cluster, because it's basically it coincides with a blue cluster. While if you add the protein level of information, this purple cluster becomes more transparent. So that's the beauty of integration. Again, you can sort of look at the patterns combining different data types and see sometimes you see something new. Another very popular in single cell currently type of unsupervised integration is through autoencoders. So autoencoder is an unsupervised neural network. Before, when I was talking about merging different omics in a hidden layer and optimizing sort of prediction of classification on the output layer, this was also a neural network, but it was supervised neural network. While autoencoder, it has this funny butterfly-looking architecture. And the idea of autoencoder, if you're not sort of into neural networks, the idea of autoencoder is to reconstruct on the output layer. You want to reconstruct your input layer. And this might sound a little bit silly. Why would you want to reconstruct your input at the output layer? But to understand this, you need to look at this bottleneck. The middle of autoencoder usually has a few nodes only, meaning that if you manage to reconstruct your input at the output by compressing your data without losing much information, you learn something fundamental about your data. So again, you are minimizing the error of reconstruction, your input at the output, by processing your data through a bottleneck, by compressing your data. And if you manage to compress your data such that you don't lose much information, then those two nodes, they capture something fundamental about your data. Those can be considered as latent factors, by the way. Maybe principal components, if it's a linear neural network. By using linear activation function, you can make autoencoder to work as a principal component analysis, for example. The reason why autoencoders are so popular in single cell RNA sequencing is because single cell analysis is essentially unsupervised. Two major methods you're using are dimension reduction and clustering. Those are unsupervised methods. Autoencoder is an unsupervised neural network. Again, you can sort of you can provide your separate omics in the as input nodes. You can sort of provide omic 1 here, omic 2 here, and omic 3 here, and merge them in the hidden layer. And this will be the integration part. Yes, so I think I am done with unsupervised lecture. This is a little bit early. So I suggest we can take questions on all three sessions, basically on all
yes.
three lectures. We can either take questions or
yeah.
I think
I will suggest the same. I will go ahead and read one that was written on HackMD that I think it's interesting for you to get started. So there was one question about limitation in terms of sample and feature number for the MOFA.
Yes, that's a good point. Actually, yes, that's MOFA. Basically, the authors of MOFA, they encourage users to do some feature pre-selection before inputting data to MOFA, because MOFA, to my experience also, it's not a very scalable algorithm. So you should try to minimize the number of features, and in order to if you want to include more samples, you should compensate it by reducing the dimensionality of your space because of technical reasons. Otherwise, MOFA will have MOFA does have technical limitations, and you should try to clean and process your data as much as possible before inputting it into Morpho.
I see one raised hand. Yes.
Yes.
Yeah, I had one question with respect to the neural network space methods that we saw some of them. Do you confirm that those methods are more adequate when we have hundreds of different observations?
So, but when you say hundreds of different, you mean the statistical observations? If you, so you're probably referring to a situation when you have a lot of data, when you have large N. So remember, we were talking about N and P.
Yeah.
So if you have sort of large N, or if your N is much greater than P, then yes, neural network will probably give you more information, or you will discover something new by using nonlinear approaches such as neural network. But you should always sort of, in order to sort of decide whether you should go for a neural network, you should always monitor your N and P dimensions. So if you're sure that for your particular multi-omics data set, you have N much greater than P, it's not always the case. But if you happen to have this limit, N much greater than P, then I would encourage you to try nonlinear methods such as neural networks.
Yeah, I'm asking because I'm working with plants, and often funds are limited. So it's not like in human where we can observe hundreds of patients. We can observe a top half a dozen of plants. So we have a lot of measurements, genes, proteins, metabolites, but number of samples is limited. So in that case, do you confirm that neural networks are probably not the best choice?
Exactly, that's the opposite situation. So indeed, when you have a lot of features, that's not something you would like to actually have when you perform data integration. So a lot of features does not mean big data. Big data is a lot of samples. So basically, neural networks are more appropriate to use when N is much greater than P.
Okay, thank you.
Yes, so we'll take Ines's question now.
Yeah, hi, thanks Nikolay for the lecture. So my question was about the autoencoders, because as far as I understood, your input layer and the output layer are going to have the same number of nodes, and therefore your integrated version of the omics is exactly like the thinnest part of the butterfly shape you mentioned. Right?
Yes, you provide the same multi-omics data set in the input and in the output.
Okay, yes, so my question was, why would you use the autoencoder then? Because since the integrated result you want is the thinnest part of the butterfly, then why use the autoencoder and not a more like classical neural network shape in which you go from a very big input layer to a very small output layer?
Yeah, so here, so I'm suggesting using an autoencoder. So it's a good way to do integration. If you want, like for single cell omics, you often want to discover populations, cell types, but you don't know them a priori. So this is what you can see. So in this bottleneck or autoencoder, you can see some new clusters that pop up after integration. So if you are using single cell RNA sequencing alone, for example, you would see 3 clusters. But if you add epigenetics information, for example, or protein level of information through autoencoder and visualize the hidden layer. So again, you plot your cells in 2-dimensional space using 2 nodes in the bottleneck of autoencoder, and you can visualize your samples here in the bottleneck, and then suddenly you can see a new cell type, which might be interesting. It might have biological relevance. So this is the idea why you are using autoencoder, because it's unsupervised. So you don't know what type of cells you discover. You don't know cell populations a priori.
Yeah. Okay. Thanks.
All right, so we are a bit on top of time. So what I will suggest is we will still take Denise's question during the break, if that's okay with you, Nikolay, and with you, Denise, so that others, if the others want to grab a water or something, they can go. What I will tell you is, let me just explain before we go to break how the next session will go. So what we will do is we will open 6 breakout rooms, and 3 of them will be for supervised integration and 3 of them for unsupervised integration. And what we will ask you all participants to do is to just split yourselves across the 6 breakout rooms as according to your interest. And then during the next 1 hour and 15 minutes, what we will do is we will be jumping from breakout room to breakout room to try to answer your questions and discuss a bit about these different methods. All right. So we will take a 8-minute break. And in the meantime, for those of you that are interested, if Denise, if you do want to go ahead and ask your question, we can stay a bit longer while the others take a break if they want.
Yes, please, guys. You can ask questions now.
I just wanted to ask about UMAP. When you mentioned that case where you want to integrate 2 data sets with UMAP, can you hear me?
Yes.
Yeah, I just want to know what you feed into UMAP when you have these 2 data matrices. You just simply concatenate them. It doesn't seem to be like maybe the best idea. So how do you do that with UMAP?
In UMAP, it's basically 3 lines of code. In the first 2 lines, so if you have two data sets, as in this example, transcriptomics and protein level of information. So first, you run UMAP on those 2 data sets separately, on RNA-seq and proteomics. And what UMAP does internally, it constructs a graph. It constructs this type of graph for 2 omics, for proteomics and RNA-seq, meaning that all cells, they know their nearest neighbors in this graph. They are connected with their nearest neighbors only. So you create 2 so-called KNN graphs with UMAP. And then on the next step, UMAP does it for you. Basically, you can overlap these 2 graphs.
So the overlapping, you do it yourself. It's not UMAP who does that.
You sort of, it's very easy. So UMAP has this option of intersecting graphs. So basically this graph intersection, which is visualized here, UMAP can do for you. So you don't, you just type 1 line of code.
So first you use UMAP to build 2 graphs and then UMAP again to overlap them.
Exactly. So you run UMAP 3 times. First time on single cell RNA-seq, second time on proteomics, and the third time when you run UMAP, there is an option of overlapping graphs.
Okay, so I didn't know about this option. I mean, I'm familiar kind of, I did use it somehow for first use case, but I didn't know about this option of overlapping 2 graphs.
It's very handy. It's very useful. It's not very well advertised actually by Leland McInnes, the creator of UMAP. It should be used more because it's super easy. So basically you can, you can use Surat and weighted nearest neighbors, for example, if you want. But essentially with UMAP, you can do integration of 2 data types with just 3 lines of code. It's super easy and super straightforward. You basically.
And so if there's more than 2 data types, then they have to be done like 2 by 2, like pairwise. If I have say 3 data sets, then
you can you can overlap multiple graphs. So as long as you constructed individual omics graphs, so you construct like here, you construct 3 individual graphs.
With
and
UMAP.
Then, of course, line of code, you actually say, I want to overlap those graphs.
Ah, okay, okay, that's cool. So you can also do multiple at once, not just two. Okay,
Yes.
that's good. Okay, that's good to know. Thank you.
You're welcome. Yes, Harun. Yeah, I see you.
Just to support the previous question, how many graphs can be merged with UMap? Maybe 200, 300?
No, this I don't know how if there is a limitation, like technical limitation, I don't know. But you typically you don't have that many omics. Yeah, you maybe have.
Yeah, actually, my question reasoning was not for omics data. I had several networks to see the merged, edges. Maybe we could use the same library to be able to just use this algorithm,
I see. No, I don't know. I guess it depends on the resources of your computer or of your cluster. I would say, to my experience, UMap is a very efficient tool, and I doubt that it can't afford merging many graphs. I would say you should try, and I believe that
Sure.
merge many graphs.
Thank you, Nikolay.
So what we will do now is we will have a small recap of both notebooks, and if there's time at the end, we can have a small discussion about different topics related with what we saw today.
Yes. Okay, welcome back. I'll open both notebooks one by one and go through major points to see what you can obtain from these benchmark data sets. So this is a schedule. I click on the HTML, and this is a supervised integration notebook on CLL data with 4 omics. And we start with exploratory data analysis. So we are supposed to have 200 individuals with chronic lymphocytic leukemia. Unfortunately, the only phenotype available for this data set was gender. That's why it's I'm using this toy phenotype. You probably can for your research you can find something maybe more interesting. But this was what I had for this data set. And we start with reading the data files, RNA sequencing data, mutations data, methylation, and drugs. There was a question about units. So the RNA-seq was originally counts, but then I double-checked. This step is not shown here, but the data was DSEQ normalized. That's why they look as floats. They don't. They are not integers. So basically, the data was normalized across samples to account for library size differences, for difference in sequence and depth. And here I immediately do some simple imputation. I think RNA-seq didn't have missing values, but in some of those 4 data sets, there were some missing values. And what I do here on the fly, I basically use median imputation, which is good enough. You can do it in a more or less advanced way. but replacing a missing value with a median value across samples for a missing feature, this is a pretty okay, simple and fast. And so methylation, I think the units were M values. So I think it was a methylation array. I think it was an epic array. So if you're familiar with this data, so it was M values, which is hard to interpret. It's not, M values are not easy to sort of to build your intuition about. But this is what is used for quantification of methylation array data. And the drugs data set, I unfortunately can't say anything about the drug data set because I always forget to check it. Like I get this question very often, but I forget to check it in the original publication, how they quantified the drug response. So it's some sort of quantification of response of each individual. So H indices, those are individual IDs, and D columns, they correspond to different drugs. So each individual responded to different drugs, different cells from each individual respond to different drugs differently. How they quantified it, I'm not sure, but I think I will post a link in the chat. I will post a link to the original publication. I think this information should be found there. So I just exited full screen in order to post the link in the chat. So next, so this is a phenotype of interest. Of course, it would be awesome to have different cancer types as a phenotype of interest, but unfortunately, all these individuals are CLL. But on the other hand, maybe there are some subtypes of CLL, and maybe it makes sense to do some unsupervised analysis on this data set in order to figure out whether we see different CLL subtypes. So for supervised integration, I used gender as a phenotype of interest. We had 79 females and 121 males. And here, what I'm doing here, I'm basically creating test and train data sets. So I'm basically here I'm selecting some fraction of samples. So 30% of samples I reserve for testing. So we are going to perform an integration, and then we are going to evaluate our model to test its performance on a test on reserved samples. So 60 samples were reserved for testing, the rest were used for training. So this is what basically I'm doing here. I'm splitting train and test samples, and some summary on test samples, so that there should be the same fraction of males in both train and test data sets. So I split train and test samples and kept the fraction between males and females in order not to end up with a situation when I by chance selected only male samples, for example. Yes, and here we start with multi-omics. So now we sort of prepared our data, split it into train and test subsets. So here I'm saying I want expression, methylation, mutation, and drugs. I want only samples that were not included in the test subset. So this is how I create a train data set. So 140 samples were used for training and 60 for testing. And this is a fundamental step in any machine learning. When you want to evaluate your model, you always separate train and test samples in order not to inflate your evaluation statistics. And so in mixomics, there is a nice filtering function, which is called near zero var. This function basically can check variance of each feature across samples and rank all your features by variance. And then what I'm doing here, I'm excluding low variant features, because if they don't vary across samples, we don't want to keep them in the analysis because they contaminate the analysis, but they are not informative. They don't provide any information that could be useful for discriminating between males and females. And so once I sort of use this near zero variance filter, I'm actually using one more filtering or feature pre-selection with lasso. So filtering and feature pre-selection, there is a number of ways. So you can do it in different ways. You can maybe skip the lasso feature pre-selection and use only near zero variance filter, or you can use both, or you can use one of them. Here I'm just demonstrating also a lasso feature pre-selection on the top. So here I'm using the GLMNet package from Rob Tipshrani and Trevor Hastie, the creators of Lasso, authors of Lasso, basically. And since we have a binary phenotype of interest, I select family binomial as information about distribution of your data, Y data. And so basically, this is a cross-validation plot. So I want to find an optimal lambda. Remember the Lagrange multiplier, the penalty coefficient. And what is important to see here is that I basically end up only with 30 or 30-something features. So this is a dramatic dimensionality reduction. So I reduced the number of dimensions from many thousands to just a few dozens. In order to fulfill in order to avoid the curse of dimensionality, basically, because we have only 200 samples. So the total number of features from all omics ideally should not be much higher than 200. And I do the same thing for methylation. So all these codes, it's basically a similar type of feature selection for methylation. And here I finally start with integration. Mixomics and Diablo, they need an input as this list of matrices, so you can visualize the dimensions of each matrix, and you see that those are not high dimensional data anymore after we did feature pre-selection. So 140 samples. So the first dimension is the number of samples. Second dimension is the number of features: 32, 10, 28, and 310. In drugs, I don't think I did any feature pre-selection because it was already good enough to work with this not very high dimensional space.
Just a quick question, can I ask?
Absolutely, yes. Go ahead.
So it's kind of weird that you have responses for so many drugs for each patient. It's not possible to treat these patients with more than 1 or 2 or 3 drugs.
I think it was like cells that were treated. So I think it's actually not a patient. So the cells, they came from different patients. indeed. So it's blood cells.
Even so, it seems it's still a pretty large number. Even if they took samples from the patients and treat them with these drugs, the number of drugs is pretty large.
300,
even if they
yes.
did that for such an experiment. So my question is, my thought is maybe drug could be a phenotype. I assume it would be very sparse, probably. So instead of gender, look at drug as phenotype. So basically look at some drugs and pick, I don't know, just a suggestion, probably would be a better phenotype. So using the three data types to predict, because this is drug response, so definitely something which would be, we could use methylation expression and mutation to predict drug response. But then again, there would be some processing and filtering and some of the drugs, how to pick the ones we want to predict. Or maybe just do it for each drug separately. I'm just saying.
Yeah, true. If you sort of have one drug, for example.
Yeah, that would be the phenotype of interest then.
Yes, that would be a replacement for Y variable.
Yeah.
Yes, so how cells respond to drug treatment. Yes. Yeah, in principle, within PLS framework, you can have matrices on both sides. So both X and Y can be matrices. So in this case, Y is not really a matrix, but a vector. But you
can, we could just feed the whole drug matrix as Y,
exactly.
like you just said.
In principle, you can do it.
Yeah, okay.
So here I so here there is an important important hyperparameter of Diablo, which is called confusion matrix. So what you see here is sort of prior guess of how well omics are related to each other. So basically, if you remember, I was showing this slide. Let me just quickly show it again. And I was explaining Diablo. So here we were correlating leading principal components from separate omics together. And we're selecting features that provide the greatest correlation between the leading principal components of individual omics. And here there was a factor C here. This is nothing else as a confusion matrix. So it's sort of prior assumption on what would be my default expectation of a relation between different omics. Since I don't have any prior assumption, this can be used, for example, you can use MOFA for getting a better guess. So MOFA can be combined with mixed omics in order to make a more proper confusion matrix. So here I just say they all are connected equally strongly. But if you know that one omic is connected, one pair of omics is especially connected compared to other ones, you can sort of play with these coefficients as a hyperparameter. So I hope it's clear. So lambda coefficient, for example, in LASSO, it's a hyperparameter, and this confusion matrix is a hyperparameter, something that you need to, some parameter above others that you have to, some model setup that you need to sort of specify prior to training, which is, this is what hyperparameter means. And this confusion matrix can be considered as a hyperparameter and optimized through cross-validation or grid search. And next, we are going, so this plot, what we are doing here, we are basically trying to figure out how many PLS components to keep in the analysis in order to get a good prediction. And this is a cross-validation plot. Again, it looks like 2 PLS components are enough, because the error is not decreasing anymore after PLS 2. So yeah, I can see that I need to move to the unsupervised notebook. So basically, here, I think the most important is to obtain this arrow consensus plot, where you visualize all the samples in a common space. And here there are a lot of arrows. So they basically mean connections between the centroid of each sample and the arrows are pointing at the positions of the samples in individual omics. So basically, maybe for clarity, you should there is a setting that you can remove all these arrows and just visualize the samples, the points. But sometimes you want to see what would be a position of the sample in expression data set alone, and how would this agree with the consensus plot. That's why people use this arrow plot. So I think all the nice visualizations here, I think these circus plots, you can appreciate the pairwise links between features from different omics and other types of networks that also can give you some hints on how features are related across omics. And also at the end, we are doing sort of prediction. We are testing our model. So once the model was trained, we use Diablo Predict. We apply it to the test data set. And here you can construct a confusion matrix to visualize what is the true value of a sample, is it's female or male, and what is the predicted value of the sample. So now I have to move to the unsupervised notebook and say a few words about unsupervised analysis. So in MOFA, there is some short introduction. So here we are, you also have 3 single cell omics for roughly 120 cells, RNA-seq, bisulfite-seq, and ATAC-seq. And I sort of do some simple filtering here. For example, I'm removing lowly expressed genes in this line. And you also can use near zero variance option from mixomics package to remove lowly variant features in order to do dimensionality reduction, in order to do some feature pre-selection, remove redundant features, non-informative features. And so those are the data distributions from the 3 data sets. And it's good to visualize them because in MOFA, you need to specify the data distributions later. This is an important step. If you don't have an idea of what data distribution you have, it would be, it might not be a good modeling then. So here I did a trick. So I binarized, for example, ATAC-seq data, because ideally it should be, ATAC-seq is an open chromatin region. It should be either covered or open. It should be just 0 and 1 values. But in real world, you still have some intermediate values. And unfortunately, those intermediate values are not accepted by MOFA. You need to, if you want to use Bernoulli distribution is an appropriate description of the binary data. But if you want to specify this Bernoulli distribution, you have to have a strictly binary data. That's why I did a trick here. I sort of binarized, I said, so all the values below 50%, I assign it to 0, and then above 50 is 1. So I kind of made this distribution more extreme. It's already pretty binary, but I made it even more extreme because here, so here when you run MOFA, you have plenty of hyperparameters to specify, but most important probably is these likelihoods. So I used Gaussian distribution for modeling expression data, although you can use Poisson, for example. Maybe Poisson distribution would be more appropriate. But I use Gaussian because I did log transformation, so Poisson becomes approximately Gaussian. And then bisulfite-seq and ATAC-seq, I use Bernoulli distribution because I binarized the data. And here another important parameter is sort of morpho is an iterative process. It drops latent factor that explain very low % of variance in the data. And the threshold is 3%. So I drop factors that explain less than 3% of variance. And that's an important hyperparameter. The rest, you can play with them, and the method is not as sensitive with respect to them. So, and as a result, you get this map, how different omics are connected within each factor. And what you can use this map for, you can, for example, look at the factor 1 and display features from RNA-seq that contribute to factor 1. And then the corresponding features contributing to factor 1 from bisulfite-seq, the CpG sites, they should have some biological link to those genes. So here you see some genes that drive factor 1 from the RNA-seq point of view. But then the corresponding CpG sites contributing to factor 1, they should be somehow connected with those genes, and this needs to be checked manually, whether it makes sense. Maybe we discover something new, maybe it's some artifact. So you need to sort of look at this relation between CpG sites and genes carefully and see how much sense it makes. But we know that RNA-seq and bisulfite-seq should be connected, should be linked in the first latent factor, etc., etc. So those are just different ways to visualize loadings, to visualize features that drive those three omics in the corresponding factors. And as a final output, you should get actually this type of plot that I demonstrated in the presentation. Unfortunately, the authors of Morpho, they sort of changed some things from Morpho 1 to Morpho 2, and this plot that I demonstrated in the presentation, it was built in Morpho 1, and they changed quite a few options in Morpho 2, and currently we are sort of switching the scores to Morpho 2, but I unfortunately didn't sort of correct all the command lines, and what you see here is not really is not really this plot, but it's sort of this plot split in 2 dimensions. So you kind of visualize your data in 1-dimensional space using factor 1 and factor 2. Sometimes you see that factor 1 allows to discriminate between the ESC cells and the rest, but factor 2 clearly discriminates between control cells and the rest cells. But those 2 factors should be plotted against each other in order to replicate this consensus plot. So again, this is a consensus plot after integration. So all 3 omics are put together here, and the samples are visualized in the consensus space. Yes, that's pretty much it. So please ask your questions.
So we are a bit over time, so we will not hold anyone that wants to leave. Some people had to leave to attend other tutorials, but we can take these 2 remaining questions. In any case, I wrote them down on the HackMD in case who asked these questions already left. So one of the questions that were, no, I see a hand raise. Okay, we'll take that first.
So I just a comment. So it seems like this MOFA package 2 does have some constraints, like in terms of specifying a data distribution. It's kind of strict, it seems. So maybe aren't the autoencoders a better option for this type of analysis nowadays?
Mm-hmm. Yeah.
Actually, this URAT package, I never worked with it, but I think it would be able to do this kind of multi-omics integration.
Yeah, so Morpho and autoencoders, it's 2 completely different unsupervised frameworks. So Morpho, in Morpho, you really model your distributions, data distributions explicitly, and join them, sort of combine them as a joint probability distribution within the Bayes rule. So you have to know probability distributions of your data in order to run MOFA.
Yes, but this seems a bit restrictive, no? It's not a bit restrictive.
Indeed. Sometimes, yes, sometimes like you have a pretty good guess if you work with counts. You're pretty sure that counts are Poisson distributed, for example, and very often we work with counts. Sometimes you have a good guess, sometimes not. Indeed, autoencoder is more flexible in this sense. You can sort of you can convert your sort of data into some non-parametric space where they forget about underlying data distributions. Mm-hmm. So you sort of you merge them to the same sort of distribution, or you transform them. You can say you transform the data to the same probability distribution, and then you merge them in the hidden layer. So indeed, those two approaches, autoencoder and MOFA, are totally different. So
have you tried to compare them on the same data set to see?
I think very often, like for this particular data set, I think I tried, and autoencoder gives you more or less similar visualization as MOFA. And yeah,
they're also probably more scalable, right? I mean, in terms of, is MOFA able to handle a few million samples?
Yeah, no. MOFA, indeed, MOFA is more high-level package where you only have a few hyperparameters, while autoencoder, usually you have to program it from scratch. There are, of course, implementations of autoencoders, but they are not really ideal. And very often when people work with neural networks, so the reason why there is no sort of a tool which is neural network which everyone can use, or very seldom you have this tool. It's because neural networks are so much customizable. So they really you really need to program a lot. You can't just use someone's neural network. Usually it's not a good idea. Well, more because it's a very low level sort of framework. Neural network is a low level software, while MOFA is a high level. You really can take a ready to use tool, specify a few hyperparameters, and run it. So I would say.
But I was also asking in terms of scalability, like with autoencoders, I know you can use few million samples, like single cells. Is MOFA able to handle a few million single cells?
I'm pretty sure not. With autoencoder, yes, indeed, you can, but it depends on implementation. I think.
Was this you remember this that we discussed a couple of days ago, Nikolay, about this news article that I shared with you? That's the author of MOFA. I think it was there was some discussion on the single cell level. So perhaps this was a very, very recent application that they did. I don't know, but I recall reading something about this application at the single cell level, wasn't it?
I would say Bayesian, yes, indeed. So just last week, or maybe like 2 days ago, there was a publication in Nature from the authors of MOFA, where it's more like a review paper where they discuss approaches for single cell multi-omics analysis, which I recommend everyone to read. Ricard Argelago and Britta Welten, the authors of MOFA. But I would say, as long as MOFA is using Bayesian framework, Bayesian framework means Markov chain Monte Carlo. So it's a very, very expensive algorithms. So so when you're talking about autoencoder, you can make it Bayesian. You can make Bayesian autoencoder, but typically people don't do it. So basically, the reason why MOFA is not really scalable is also because of the Bayesian type of analysis, which is sort of notoriously slow. It's based on sampling, but on the other hand, it's more robust, maybe.
Okay, thank you.
Kerem.
Yeah, actually, I've noticed also on the chat, they gave the same question. What can you tell about the interpretability of this outcome? I mean, how do you interpret it for biologists? What happened and what is the main concern?
So are you asking about some particular method now? Interpretability of autoencoder?
Let's say for MOFA, I mean, I have the same problem with principal components, but here you have something else called factor something.
Factor analysis, yeah.
How do you interpret what changed?
Mm-hmm. Yeah, so basically about interpretability, I think as long as any machine learning algorithm can always, including morpher or autoencoder or neural network, it can always deliver you key drivers in your biological process. So it's basically a way to understand out of 20,000 genes, what genes should I look at? It's a way to find candidates for you. And any machine learning framework can give these candidates to you. This is interpretability. This is sort of if some algorithm predicts something. So we were talking about prediction as a criterion of successful integration. If an algorithm can predict, you can always roll it back and understand why it predicts. You can understand what kind of features, genes, proteins, etc., what kind of features they drive this good prediction. And this is pretty much, it's pretty lot of information. It gives you sort of some feeling. What should I look? What should I look at? What should I? What features should I pay particular attention to? So it's a way to sort of rank. So ranking is an interpretation. So regarding how to explain principal component analysis and factor analysis to a biologist, those are actually quite easy to explain compared to neural network. Because principal components analysis, it's a linear algebra, it's sort of more understandable thing. Well, neural network, then it becomes really trouble to understand how to explain it to a biologist. But you know, just to make it simple, principal component analysis is a way to again, it's a way to understand what are the most important features, what are the most important genes in your data set, because you have too many. And like one of the goals of bioinformatics is go from many candidates to one or two candidates. Then it will be a lot of information for a biologist, I would say.
There was a question on the chat as well that you've, I believe you partially addressed, but I don't know, maybe you want to add something more. So the question was, any special considerations to combine binary data versus non-binary data in integration?
Yeah, yes. So right. So it's indeed it's non-trivial, and it's basically the general problem in omics integration that you have data coming from different distributions. How to combine them correctly? It's the same type of problem. How to combine binary and non-binary data? Those are just two data types coming from different probability distributions. So. So either you model these distributions explicitly, like Morpho does, so you know that it's a binary data, you know that it's a Bernoulli distribution, or you convert your data. If you're not sure about the probability distribution of your data, you try to convert your data to graph, for example, to some non-parametric space where you keep only between individual links, but you sort of remove all other technological information. So the technology, the omics specific type of information disappears, and you keep something which is shared across different data types.